What's up, Ninja Nerds? In this video today, we're going to be talking about Hodgkin's lymphoma. Before we get started, if you guys like this video, it makes sense for you, please support us. One of the best ways that you guys truly can do that is by hitting that like button, commenting down in the comment section, and please subscribe. Also, do you want some amazing notes and illustrations that help to follow along with me during this process? Go down the description box below. We got a link to our website where you guys can check all that stuff out. But starting off with Hodgkin's lymphoma. Hodgkin's lymphoma is a very interesting disease, and really it comes down to a very in-depth lymphocyte pathway alteration. So there's an alteration or an abnormality that develops within the lymphocyte pathway. Now, I don't want us to go crazy into this pathway. I want us to have a basic understanding of the pathway and understand where along the pathway there's disruption or alteration. So let's quickly go over the lymphocyte pathway. And what I want to start off with is, what are the organs involved? What is the pathway involved? Maybe what are some specific intermediaries that are involved? Let's talk about that a little bit. So the first part here that we have to discuss is we start all of hematopoiesis off in what's called the red bone marrow. So hematopoiesis is the production of all blood cells. That includes lymphocytes. Now, in the red bone marrow, what we start off with is what's called a hemocytoblast. And a hemocytoblast should differentiate into what's called a myeloid and lymphoid stem cell. Which is the stem cell that's involved in the production of lymphocytes? The lymphoid stem cell. So that's the one that I really care about. So let me have right here, this is going to be our lymphoid stem cell. That's the precursor for basically all your lymphocytes. And what it does is it makes what's called precursor B cells and precursor T cells. So what I'm going to push out here into the bloodstream is a precursor T cell. And then this precursor T cell will then move to the thymus. And then at the thymus, it'll undergo a little bit more differentiation. So what I'm going to put is I'm going to put this as a precursor T cell. And then I'm going to push this into the bloodstream. And this is a precursor B cell. And the precursor B cell should move to the lymph node, right? And the specific part of the lymph node, I'll diagram out which part of the lymph node we're actually looking at in a second. But this is the process. So we have the bone marrow making precursor B cells and precursor T cells. The precursor T cell should go to the thymus. The reason why is in the thymus it should undergo a little bit more development, maturation, and then differentiation into two specific types of T cells. And these particular T cells are different based upon the specific type of cluster differentiation protein they express. So one I'll draw here in like this pink color. So it's got these specific types of cluster differentiation proteins. And then another one I'll draw here in like this bluish color here. This is another cell with cluster differentiation proteins, but it's a different type. So these are called your CD4 proteins. So these are your CD4 positive cells. So what happens is the precursor T cell, which doesn't know what he's supposed to be, goes to the thymus and develops CD4 positive T cells. These are also called T helper cells. And then the other one has CD8 positive proteins. So he doesn't know what kind of cell he's supposed to be, goes to the thymus, differentiates into CD8 positive cells. These are also called T cytotoxic T cells. Now, that's the process of T cell development. The problem is that not many Hodgkin's lymphomas, really any, present with any T cell Hodgkin's lymphoma. It's primarily B cells. So let's take our time and focus on this pathway. What I'm doing is I'm zooming in on what's called a lymph node, like the germinal center of it. So here we have the germinal center, and here's the pre-germinal center. If you guys want to understand what exactly I'm referring to, imagine here I draw a small lymph node. Here's the efferent lymphatic vessels, and you have a bunch of afferent lymphatic vessels going into it, right? What I'm really looking at is you have these things called trabeculae, which separate out the lymph node, and then we're looking at these like little follicle parts here. This is really what we're zooming in on right here, this part here. So let's actually talk a little bit about this. I don't want to go crazy. We'll focus on this a little bit more in non-Hodgkin's lymphoma. But what happens is this precursor B cell, if you want to think about it, it's also naive. So we can also call this not only a precursor, but we can also call it naive B cell, meaning it has never been exposed to an antigen. So at this point, when it's a naive B cell or a precursor B cell that's naive, it has no antigen exposure. What happens is it goes to the actual lymph node and it moves into this area here called the mantle zone. And in the mantle zone is the zone of antigen exposure. It could also occur in the blood, but we're going to say that this is the area of antigen exposure for simplicity's sake. So now here what happens is, this is going to be that we're going to move these precursor or naive B cells into the mantle zone. And some of these will actually become exposed to antigens. Some of them will not become exposed to antigens. The ones that do not become exposed to antigens stay in the mantle zone. The ones that do become exposed to antigens, they move into the germinal center. So now what I'm going to do is I'm going to represent this antigen. Let's say on this B cell surface it has little receptors. It's called B cell receptors. And when it gets exposed to a particular antigen, so we'll make this antigen, let's make it red. Here's this antigen. So we're getting exposed to a antigen. I'm going to write this as a antigen. This is a antigen that we're getting exposed to. The B cell, once it gets recognized, it binds to the actual B cell receptor, it takes it in. And then once it does that, it exposes a piece of it on its MHC2 complex. We'll get into that in a second, but it takes it in, it gets exposed to an antigen. That's the big key here. Once it gets exposed to a big antigen, it becomes a special type of cell. And this cell is very interesting in the sense that it's a really large cell. A really large cell, kind of a bunch of different nuclei on it. And it's been exposed to this particular antigen. What happens is, this is called a centroblast. This is called a centroblast. Now the centroblast, what it's supposed to do is it'll take this antigen, right, that it bound to, and it's supposed to then have a very strong affinity for antigens very similarly to this. What happens is there's a kind of a pathway here where you diverge. One is if the affinity that these B cell receptors have for that antigen is weak. So we say a decreased affinity for antigens from this B cell receptor, then these are supposed to undergo apoptosis. These cells should die. That's naturally what should happen, is that these cells should undergo apoptosis. Because if you don't have a strong desire or a strong affinity for antigens, then what kind of purpose do you have, B cells? You don't have a purpose. Because again, the B cell receptor is supposed to be a very good, strong antigen attachment structure. So when it sees antigen, it should have a strong affinity and bind to those antigens. If it doesn't have a very good affinity for antigens, it's not a good cell. We should actually kill it and not let it be there because it can cause disease. Or it doesn't have any effect. But if it does have a strong affinity, if the affinity is strong, it has a good affinity for the antigens, then what it does, it moves down and it moves into something else, another cell. And this cell is called a centrocyte. This is called a centrocyte. And now, here's what I want you guys to understand. The centrocytes is just a little bit more mature, a little bit more developed. So it's got these B cell receptors which are primed, they're ready, they're really good at their job. Okay, they're really good at their job. They're good at being able to identify antigens. But you know what B cells are also supposed to become? They're also supposed to become plasma cells. So you can have two different types of B cells. One is a memory B cell, means it has these B cell receptors which can identify antigen for years and years and years. It's good at that. But you should also have a type of cell that can produce antibodies that can also recognize that antigen. So we should have two different types of cells, a plasma cell and a memory B cell. So with that being the case, these centrocytes become exposed to two other cells in this lymphoid kind of lymph node structure. This cell here is called a T cell. So this one here is called a T cell. This is a T cell. And this one here is called a dendritic cell. This is called a dendritic cell. Now what happens is the dendritic cell has a very specific antigen that it exposes. And we should already know that this antigen will be very nicely recognized by that B cell receptor. So the B cell receptor will be able to recognize the antigen on the actual dendritic cell. But the other thing is T cells have a very special type of molecule on their surface. They have like this MHC2 molecule. They have this little MHC2 molecule that can also expose an antigen. And this should also be able to interact with the centrocyte. When the centrocyte interacts with these things, it then from that point decides what it's going to do. All right? So then what happens is, if it's decided, okay, I like interacting with antigens and being a memory B cell, it'll become a memory B cell. Oh, I like becoming a plasma cell and producing antibodies, it'll become a plasma cell. So at this site right here, it undergoes a process called class switching. So from here, when it interacts with these two, it'll move right up here into something called the marginal zone, into the marginal zone. And then from this point here, this centrocyte is differentiated and it says, okay, I know what I'm going to be. From here, I'm going to become a memory B cell, or I'm going to become a plasmoblast, which then actually becomes a plasma cell. So what are the two different things that happen here? So at this point, the centrocyte, once it goes through this pathway right here, what is this? What happens here? At this step here, it undergoes something called class switching, where it has the ability to produce different antibodies. That's all it means. It switches its antibody production from maybe IgM to IgG and IgA, right? So that's the whole point. So it undergoes class switching. And then it moves into the marginal zone. Once it's in the marginal zone, it then differentiates and decides what it's going to become. So at this point, if it goes this pathway and expresses tons of these B cell receptors which are good at recognizing antigens, this is a memory B cell. If it goes this way, which is going to be leading to the actual formation of plasma cells, which are cells that produce amazing antibodies, then this is going to be going in the direction of the plasma cell. So this is called a memory B cell, and this is called a plasmoblast, which then becomes a plasma cell, which produces antibodies. And what do these antibodies recognize? These antibodies are supposed to recognize those antigens freely circulating. These memory B cells are supposed to recognize any of these antigens freely circulating. That's the cool concept of these things. So now the question you probably have is, okay, Zach, that's a lot of cool stuff, but where the heck is Hodgkin's lymphoma involved? Hodgkin's lymphoma is involved kind of like somewhere in this process here. And so what's the thought process here is that something is happening where centroblasts, which are supposed to undergo apoptosis, do not undergo apoptosis. And so what happens is these centroblasts, they never actually completely undergo apoptosis. So this is where it actually happens. This is where the disease process occurs. And if the centroblasts never undergo apoptosis, they continue to keep kind of like replicating, but they never fully differentiate. So that's the other problem. They never fully differentiate, and they kind of just stay in this stage where you have tons of these centroblasts. But these centroblasts look a little bit odd. They don't actually have this characteristic shape like you would think. They take on this weird kind of shape. So what happens is you get a ton of these centroblasts, but these centroblasts take on a very odd type of shape. And you know what this actual shape is? Let me show you here. What happens is they kind of look a little bit like this. They have a big nuclei here and a big nuclei here that kind of looks like two eyes. And we call these cells Reed-Sternberg cells. So these are called Reed-Sternberg cells. And that's the problem. You get a bunch of these. And what's the reason why you get a bunch of these stuck in the lymph node? It's because the cells won't undergo apoptosis and they won't completely kind of differentiate. And so you get a bunch of these like owl eye looking cells that accumulate within the lymph node. Now the question then comes, what is the reason why these cells don't undergo apoptosis? That's a great question. Let's come down and talk about the reasons as to why that happens. The big point here is that we got these cells, right? So we talked about how you have your B cell, how it's naive, goes into the mantle zone, becomes exposed to an antigen. If it gets exposed to an antigen, it becomes a centroblast. The centroblast will then have a very strong affinity. It'll become a centrocyte. The centrocyte, after it undergoes, so there's a specific process here where you go naive B cell, antigen exposure to a centroblast. Centroblast, if it has a very strong affinity, there's something it does. It actually switches the variable region of its actual B cell receptor. It's called somatic hypermutation. So we'll actually quickly write that. It's called somatic hypermutation. somatic hypermutation. That's for the centroblast. Then it'll go and become a centrocyte. The centrocyte undergoes a very special process called class switching. And then from there, it'll move into the marginal zone and give way to memory B cells, or give way to plasmablasts, which become plasma cells. So that was the basic kind of process here. Now, what happens is we said that technically at some point here, these centroblasts were maybe some of them that didn't have that very good somatic hypermutation, or they didn't have that strong affinity. The decreased affinity was the problem, right? They go this way. Whereas the ones that have very good affinity for the antigens, they go this way and they continue to differentiate. And what's supposed to happen to these cells is they're supposed to undergo apoptosis. But it doesn't happen. So the question then arises, like, how does this actually happen? Well, it comes down to like the actual like genetic material in these lymphocytes. So let's say we zoom in on these actual lymphocytes. We're zooming in on one of the lymphocytes. Here we're gonna have the DNA. And there's different types of genes that are kind of involved here. The one gene that's really been thought to be heavily involved here is called nuclear factor kappa beta. So it produces a molecule called nuclear factor kappa beta. So this produces a molecule called nuclear factor kappa beta. What nuclear factor kappa beta does is you know, in order for cells to kind of divide, they have different processes. You have what's called proto-oncogenes, and these are genes that want to have a lot of cellular proliferation. So we have two different types of genes here. One is we have what's called proto-oncogenes, and then you have what's called apoptosis genes. So in proto-oncogenes, in these situations, this isn't the problem. But if they were on, what they would do is these help to cause increase in cell proliferation. All right, cell proliferation. If these are hyperactive, if you have what's called anti-apoptotic genes, so they don't want to allow for apoptosis, then we have what's called anti-apoptosis genes. These will basically decrease cell death. So then the cells won't die. Either way, in both of these scenarios, the cells continue to survive. So what happens is the nuclear factor kappa beta will basically go over here and particularly turn off some of these actual genes. And so what it'll do is you'll take these anti-apoptosis genes, right, which you basically, if these are kind of like on, they'll basically prevent the cell from ever being able to die. And so that's what it does, is it kind of goes over and tells like these apoptosis genes, hey, go ahead and turn off. So if we turn off apoptotic genes, or we decrease the apoptotic genes, then the cells won't die. And so that's basically what happens. It's the same kind of effect here. And so basically what we're doing is we're turning off apoptosis. And if I decrease the apoptosis, this is the basic concept. The cells will not die. There's no problem here with the proto-oncogenes. It's really the cells just refuse to die. And so if they don't die, they continue to survive and survive and survive, and you should get a lot of these Reed-Sternberg cells that just accumulate within the lymph nodes. And that's really where the problem comes. But the question is what actually causes this upregulation? So there's a lot of this what's called nuclear factor kappa beta. What's the reason for this? There's not a great answer, but what's thought is that nuclear factor kappa beta may be in these high amounts secondary to what's called Epstein-Barr virus, which is the virus that causes infectious mononucleosis. And it may be secondary to things like HIV or immunosuppressed states. But again, the mechanism exactly of how it does that, we're not sure. But what we do know is that the nuclear factor kappa beta will decrease apoptosis. And if you decrease apoptosis, you decrease cell death. And so that causes these Reed-Sternberg cells to continue to accumulate in the actual lymph nodes and cause disease processes. So here's the recap. Lymphocytes are made in the red bone marrow, the precursor ones. They get into the bloodstream, particularly the B cell type. It should then go to the lymph node. As it goes to the lymph node, it gets exposed into antigens in the mantle zone. If it gets exposed, it then goes into the germinal center, becoming centroblasts. Centroblasts now have that B cell receptor, which it bound to the antigen. It should actually somatic hypermutation. In other words, it should change the antibody structure just a little bit to be able to recognize variable antigens. Once it does that, if it has a good affinity, it continues to go to centrocytes. If it does actually go to centrocytes, then it'll undergo class switching. It'll then become a little bit more differentiated into either a memory B cell or a plasma cell. The breakdown in Hodgkin's lymphoma is really that the cells refuse to undergo apoptosis because of their poor affinity. And they refuse to completely differentiate and become specific cells. So they kind of get stuck into this intermediate of like a centroblast, but looks weird, like a Reed-Sternberg cell. And the reason why this happens is you've turned off the apoptotic genes because of an increase in nuclear factor kappa beta, which may be related to viruses such as Epstein-Barr viruses or HIV. Okay, the question then comes, what would it look like if you get a lot of these weird funky cells, these Reed-Sternberg cells? Let's actually draw this. One of these funky like little looking Reed-Sternberg cells that are accumulating inside of the actual lymph node. Let's talk about that next. All right, so now we got a lot of these Reed-Sternberg cells just accumulating like rabbits in the lymph nodes. And again, the reason why is there was a breakdown in the apoptosis pathway. So when the lymphocytes are supposed to undergo apoptosis, they don't. And the reason why may be due to Epstein-Barr virus, this may be HIV, this could be immunosuppressed states in general, like after they get a transplant or they're on chemotherapy, whatever it may be. But it causes this increase in nuclear factor kappa beta, which kind of shuts off the apoptosis gene, so they continue to keep thriving. And it's kind of like these weird like intermediaries of centroblasts, which are called Reed-Sternberg cells. Now, if you get a lot of these Reed-Sternberg cells that accumulate in the actual lymph nodes, what does this look like? Well, here, let's say that we get a bunch of these suckers just accumulating in the actual lymph nodes because they just refuse to die. They refuse to die. If they refuse to die and they continue to kind of thrive within this lymph node, then what happens is you get something called lymphadenopathy. And this is really a problematic issue for these patients. So lymphadenopathy kind of occurs when you get like these different lymph nodes that get kind of big and swollen and enlarged. And so that's really the problem with this, is that this can occur all over the dang place. Where can this occur? When you get lymphadenopathy, the most common areas tend to occur in the cervical region, the supraclavicular region, the axillary region, and then one other region. So they can occur on both sides though. Cervical, they can occur supraclavicular, they can occur in axillary. One other area which is kind of an interesting one, and that is mediastinal. They can also occur in the mediastinal lymph nodes. But one of the huge things here is, with the lymphadenopathy, it's usually painless. It's a painless lymphadenopathy. They're oftentimes slightly rubbery in their actual texture when you feel them. And there's one more big feature here. It's contiguous. So it's contiguous or continuous. What does that mean? In other words, let's say here that this is the lymph node that gets actually a lot of these Reed-Sternberg cells accumulating. If it accumulates in this lymph node, it'll travel to this one, to this one, to this one, to this one, to this one. It's a straight shot that it continues to go in the order of lymph nodes in the proximal vicinity. That is super important. But again, lymphadenopathy is going to be one of the key features here. The questions again come as which areas, so let's write these down again. One of them is going to be cervical lymphadenopathy. The other one is supraclavicular is another really big one. Another one is axillary. And the last one that I really want you to remember here is mediastinal. Mediastinal. And the reason why is the mediastinal is very common sometimes in the patients with Hodgkin's lymphoma. And it can present a lot of different ways. So think about it. It's in the proximity of a lot of different structures here. So it can definitely cause irritation or agitation or compression of structures like the heart, the lungs, or the superior vena cava. So watch out for things like cough and shortness of breath and chest pain. And the reason why is this may be compressing on nearby structures. So this can cause a lot of different things. So it can cause maybe some of the cough and the shortness of breath due to compression of the lungs. It can cause chest pain because of compression of the pericardium or the pleura. And it also can cause something like superior vena cava syndrome due to compression of the superior vena cava. So watch out for any kind of like edema or bluish discoloration of the neck and the face and the arms. So that's really big things to watch out for here. Now, we know lymphadenopathy is key. It's because these Reed-Sternberg cells are accumulating in the lymph nodes and they're just refusing to die because of the increase in nuclear factor kappa beta. The other problem is is that these dang kind of like Reed-Sternberg cells, they like to secrete a lot of different types of cytokines. And this mechanism is believed to be one of the reasons for a lot of different things. So one is they can release things like interleukin 1, interleukin 6, tumor necrotic factor alpha, a bunch of different things. But what this is thought to do is these cytokines are thought to go and stimulate multiple different tissues. And one of the tissues is the hypothalamus. So it may act on the hypothalamus, and the hypothalamus is really, really sensitive to these cytokines because what it'll do is it'll actually increase the body's thermostat because it's kind of like an inflammatory signal. So they try to increase the body's temperature. So this can lead to fevers. This can lead to night sweats. And what's really interesting about this is that these symptoms oftentimes occur very sporadically. And this is a very interesting term. So we have, first off, lymphadenopathy is a really, really big feature here. But the other one here is the fevers and the night sweats. This kind of stuff can happen really it's like sporadically, and we call this B symptoms. B symptoms. And so this is a really, really important thing to remember is that you get these kind of like periodic kind of symptoms here where they can have like a fevers and night sweats and maybe even some degree of weight loss. They may even have some degree of weight loss. So this can happen kind of continuously or it can occur periodically. That's another important thing to remember. This is usually continuous if it's B symptoms, but if it occurs periodically, where they have these like high fevers and night sweats for like maybe a couple weeks, and then they become afebrile and no night sweats for a couple weeks, and they go back, it's a periodic, it's a cyclic process. They call that Pell-Epstein fever. So remember that there is a variant of this. There's a variant of this B symptoms, which is you can have a cyclic, let's just call it cyclic B symptoms. If it's cyclic B symptoms, we call this Pell-Epstein fevers. Pell-Epstein fevers. This is more of a cyclical process of these fevers. So just remember that as well, okay? So far, we have lymphadenopathy in these particular areas that is contiguous, painless, rubbery. We have B symptoms that can be continuous or periodic Pell-Epstein fevers. Another thing that can happen here, which is really weird, it's not as specific. It's actually it's not very common, but it's highly specific for Hodgkin's lymphoma. Pell-Epstein fever, but there's another one. Sometimes the lymphadenopathy can actually be painful, which the mechanism is not completely understood whenever the patient drinks alcohol. So there's a really thing to add, an additional thing to add on here. So sometimes you can get what's called a alcohol-induced lymph node pain. And this is very rare, but highly specific for Hodgkin's lymphoma. So if you see this on your exam, don't forget this one, okay? Same thing like the Pell-Epstein fevers. They're very rare, but they're highly specific for Hodgkin's lymphoma. All right, what else with the cytokines? The cytokines can also tell the liver to produce things like an increase in the erythrocyte sedimentation rate, or an increase in the CRP. So these may be lab markers that you could check to see that there's a lot of markers of this liver reactivity. It also tells the bone marrow, and the bone marrow responds to a lot of these inflammatory cytokines by decreasing the number of red blood cells and increasing the number of eosinophils. So what is the effect here? For some weird reason, you can see this anemia of chronic disease where you drop your red blood cells, but you see an eosinophilia because you increase your eosinophils. So these are other kind of weird things to be able to take into consideration as well. But the big thing I want you to remember is lymphadenopathy that is contiguous. You may see B symptoms due to these cytokines causing the hypothalamus to upregulate the body temperature, causing fevers and night sweats and maybe some degree of weight loss. If it's alcohol-induced pain or Pell-Epstein fevers, they're highly specific for Hodgkin's lymphoma, but very rare. You may see acute phase reactive proteins. You may see some degree of anemia, particularly anemia of chronic disease, really. This should actually be more specifically anemia of chronic disease. And this is eosinophilia. The last particular situation is another one that's highly specific for Hodgkin's lymphoma, but very rare. This is another one where you can get a lot of these like basophils, and these basophils can become hyperactivated, And what they do is they increase, so the basophils, they basically increase their histamine release. And if you increase histamine release, what this does is this produces something called pruritus. This produces something called pruritus, so itching of the skin. This produces like a local inflammatory reaction, if you will. And this is another thing that I want you guys to remember. So the big things to take away at this point before we move on to the next part, which is extranodal tissues, is you're going to get lymph node involvement due to the increase in Reed-Sternberg cells. That's going to be causing lymphadenopathy. It's contiguous. If they release cytokines, the cytokines can cause a bunch of different things such as B symptoms. If it's periodic, it's Pell-Epstein fever. If when they drink alcohol it causes lymph node pain, it's highly specific but rare, seen in Hodgkin's lymphoma. You may also see evidence of acute phase reactive proteins being released. You may see evidence of anemia, chronic disease, and eosinophilia. You may also see pruritus due to lots of basophils releasing histamines. Another thing that you could potentially see here, and this is a controversial topic, is you could see spleen involvement. And the reason why this is controversial is because sometimes there's what's called extranodal tissue involvement and lymph node tissue involvement, which you can see in lymphomas. In Hodgkin's lymphoma, technically, the spleen is considered, in many different textbooks, it's considered to be a part of nodal tissue. So technically, if we're really being specific, this is a nodal tissue. So we can't consider this to be a part of the extranodal tissue involvement. So what can happen here? Sometimes these lymph nodes can actually, these lymphocytes, these Reed-Sternberg cells can populate within the spleen. And if they populate within the spleen, they can cause enlargement of the spleen and cause splenomegaly. And if they cause splenomegaly, they may cause nausea, vomiting. They may cause anorexia. They may cause left upper quadrant pain. And the reason why is as you enlarge this structure, you compress on things like the stomach. The stomach is right next to it. And if you compress on the stomach, you make less room. As the patients eat, they get full really quickly. They kind of get nauseous. They don't want to eat a lot. And that's another feature that you want to watch out for, is splenomegaly. But again, big thing is it's controversial because some people say, oh, it should be an extranodal tissue, but the textbooks have kind of stuck with it being technically a nodal tissue, so we can't consider that. The last thing here is, with Hodgkin's lymphoma, it is very rare for you to get extranodal involvement, but we need to talk about it because we should understand that this is a possibility, it's just not as common. This is more common in non-Hodgkin's lymphoma. That's the big difference. But, so we are thorough here, what could this potentially look like? It's very straightforward. We said that the Reed-Sternberg cells are causing a lot of these, they're just accumulating inside of the lymph nodes. They're populating like bunnies. And so you get these big kind of lymph nodes, lymphadenopathy that we talked about, but sometimes they can actually seed into the bloodstream. And if they seed into the bloodstream, they can spread to other tissues. So if it spreads to the GIT, it can cause a bunch of different problems here. So we'll talk about this a little bit later, but it can spread to the GIT and cause a lot of different symptoms here. It could potentially spread to the central nervous system. It could potentially spread to the liver and cause hepatomegaly. It could potentially spread to the glands like the salivary glands, the thyroid gland, the lacrimal glands. It could potentially spread to the bone marrow. So you can see CNS, you can see liver, you can see glands, you can see bone marrow. It could even spread to the pleura and the pericardium. So it could spread to the pleura and the pericardium. Or it could even spread to the skin. However, these are not as common as you will see, and it can be way more common in non-Hodgkin's lymphoma. So the big thing I want you to remember here is it's more common in non-Hodgkin's lymphoma. And that's a really, really important thing here. You can potentially see it, it is just not very common. And the symptoms that they will have are dependent upon the tissue that they deposit in. So if they deposit it into the GI tissue, they may cause things like bowel obstruction, they may cause things like abdominal pain. If they deposit into the central nervous system, they cause things like seizures, they may cause tumors. It can cause hepatomegaly. It can cause maybe decreased salivary and lacrimal gland production, dry eyes, dry mouth, thyroid enlargement. If it deposits into the bone marrow, it can decrease other cell production. So it may cause pancytopenia and bone pain. If it deposits to the skin, it can cause skin lesions. If it deposits into the pleural and pericardium, it can cause things like cardiac tamponade and pleural effusions. So there's a lot of different things that you can see here. It's just important for you to remember, and I think this is really, really key here. This is way more common in non-Hodgkin's lymphoma, but it's still slightly possible. Now, at this point, we have covered Hodgkin's lymphoma, how it actually develops. We covered the causes. We've covered what are the effects of having a lot of these like Reed-Sternberg cells all over the body. Now let's talk about how do we diagnose Hodgkin's lymphoma. All right, my friends, so we have a patient who comes in, they have lymphadenopathy as their primary symptom, right? So they come in, they have some lymphadenopathy, maybe it's cervical, maybe it's axillary, maybe it's supraclavicular, maybe they have some compressive symptoms from mediastinal lymphadenopathy like cough or shortness of breath due to compression of the structures like the pleura and the trachea and things to that effect, or maybe they have chest pain due to actually involvement or compression of the heart, the pericardium, maybe they have superior vena cava syndrome due to compression of the SVC. So you get the point, they can have a lot of those features. If they also come in with B symptoms, so fevers, night sweats, weight loss, right? Maybe due to the cytokine storm. If they also have it periodically, which is Hell-Epstein fevers, or if they have alcohol-induced lymph node pain, or if they have pruritus, these can be highly specific but rare in Hodgkin's lymphoma. Also, watch out for any spleen involvement like splenomegaly. This may cause early nausea, vomiting, abdominal pain, abdominal fullness, that effect. And remember, extranodal involvement is possible, it's just nowhere near as common as patients who have non-Hodgkin's lymphoma. That is way more common and easier to remember for your exams, but it is possible. If that happens, especially the contiguous lymphadenopathy, you should start right off and say, okay, I just need to find those Reed-Sternberg cells. If I find the Reed-Sternberg cells, then we're good. Problem is that you can have lymph nodes that are big and puffy, and it could be maybe Hodgkin's, could be non-Hodgkin's, or it could be metastasis. How do I know which one it is? Biopsy it and look for the Reed-Sternberg cells. So that's the first purpose of the lymph node biopsy, is here you have this mass. I want to know, is it Mets versus lymphoma? So when I take this piece of actual tissue, what I'm going to be looking for is what? I'm going to be looking for these goofy looking things. What are these called again? My Reed-Sternberg cells. So the first thing I'm going to do is I want to be able to find a lot of these. So my first question is it Mets versus lymphoma? In other words, is it a cancer from somewhere else that spread to a lymph node? Because that's possible. And is it Hodgkin versus non-Hodgkin's? So first thing I do is I biopsy that actual area. Once I biopsy it, if I see Reed-Sternberg cells, or, gotta be very, very careful, Reed-Sternberg cells or a variant called popcorn cells. So these are basically a Reed-Sternberg variant. They are kind of pretty much equivalent to them. If that is present, right, if these are positive, then you have Hodgkin's lymphoma. That is really what it comes down to, my friends. So you start off with the actual, okay, is it Mets versus lymphoma? Biopsy it. Once you biopsy it, and you basically say, okay, once I biopsy, I can say no cancer cells, right? No, let's actually be more specific because technically Hodgkin's lymphoma is a cancer. No Mets. And then once I see here, I go to the next part, which is, okay, I find Reed-Sternberg cells. If there is no Reed-Sternberg cells, this is a possibility of being non-Hodgkin's lymphoma, right? So that's a really, really important thing. So we start off with a lymph node biopsy, and what we're trying to be able to find is there Reed-Sternberg cells or is there popcorn cells? Now, once we found Reed-Sternberg cells or the popcorn cells, we have a pretty good suggestion that this is Hodgkin's lymphoma. So then the question comes, okay, if it is Hodgkin's lymphoma, how can I really determine? So the next kind of question comes is, how do I know if I have these Reed-Sternberg cells, these little owl-eye looking things, or these other cells that are kind of like it, but it's called popcorn cells. So now the next question is it Reed-Sternberg cells or is it popcorn cells? Because I know that, yes, this is both present in Hodgkin's lymphoma, but I need to know which one is it. So then I go to the next test, my flow cytometry. Flow cytometry will basically say, okay, I did my biopsy. I found Reed-Sternberg cells, or I found popcorn cells, okay, which are basically a variant of Reed-Sternberg cells. So Reed-Sternberg popcorn cells. The difference and nice thing between these here is that they have differences in their proteins they expressed on their surface. So you guys know by this point, after watching all the leukemia videos, that flow cytometry is great at being able to recognize specific proteins on the cell surface. Because what we can do is we can make antibodies that are basically fluorescent and tag these specific proteins. And so what I'm tagging here in the Reed-Sternberg cell is CD15 and CD30. Let me make this bigger so you guys can see this. So CD15 and CD30. For the popcorn cells, it's CD20 and CD45. If the CD15 is positive and the CD30 is positive, we call this classical Hodgkin's lymphoma. So we're going to abbreviate this, classical Hodgkin's lymphoma. Okay? And there's 4 different subtypes from that one. You're like, what the heck? Why doesn't it end? If this is positive, the CD20 is positive, and the CD45 is positive, this is called non-classical Hodgkin's lymphoma. I mean, non-classical Hodgkin's lymphoma. And there's only one subtype for this one, thank goodness. Now the big thing is that classical Hodgkin's lymphoma is much more common. Non-classical Hodgkin's lymphoma is much less common. So that's another really big thing to take away from here. This is more common, this is less common. All right, so at this point, what I want you guys to understand then is that if we've gone through, we said, hey, we got a big old lymph node, I biopsy it. My question is it a MET? Is it Hodgkin's, non-Hodgkin's? From that biopsy, I'll be able to determine, oh, there's no METs. Good. Is there any Reed-Sternberg cells? Oh, there is Reed-Sternberg cells, or there's a variant of them, the popcorn cells. Oh, well, if that's the case, that's possibly Hodgkin's lymphoma. There's no Reed-Sternberg cells. Oh, that could be non-Hodgkin's. We'll talk about that in a non-Hodgkin's lecture. If I see Reed-Sternberg cells or popcorn cells, I have a pretty strong suspicion that this is Hodgkin's lymphoma. So then how do I determine which subtype of Hodgkin's lymphoma it is, classical or non-classical? I go to flow cytometry, and I find which one it is. If it's Reed-Sternberg cell positive, it's classical Hodgkin's lymphoma. That's the most common type. If I find popcorn cells or the Reed-Sternberg variant, that's the non-classical Hodgkin's lymphoma. That's the less common type. Now, at this point, then the next question is, there's subtypes of this one and there's one subtype of this one. How do I know which one it is? Let's come down for a second. So I go to the next part here, right? Either I'm Reed-Sternberg cell positive or I'm popcorn cell positive, or if you want to even be more detailed, just to recap, I'm CD15, 30 positive, or I'm CD20, 45 positive. This is all the classicals. This is non-classical. For the classical, there's 4 subtypes, and we can identify these based upon their histopathological appearance. I'll show you guys each one at a time. The most common one is called nodular sclerosis. This is going to be the most common type. It looks like this. The next one here is called mixed cellularity. This is called mixed cellularity. This is the second most common. So this is the most common. This is the second most common. And it looks like this. The third one is called lymphocyte rich. So lymphocyte rich, and the nice thing about this one is that this has the best prognosis. So one of the benefits of doing this test, this histopathology, is you want to be able to find which one has a good prognosis. And then again, lymphocyte rich looks like this. And then you also want to be able to identify the poor prognosis, the lymphocyte depleted. Thank goodness this is one of the least common, but it has the poor prognosis or the worst prognosis. And it looks like this. So it's important for you to be able to realize the classical Hodgkin's lymphoma with the most common type being nodular sclerosis. But it's also important for you to be able to recognize the one with the best prognosis and the worst prognosis. Now, it shouldn't be something that you're expected to be tested on off the histopathology. It's just something to give you guys an idea of what this looks like under the microscope. Now, the other one is the non-classical Hodgkin's lymphoma. If this is the case, there's only one type, and it's called nodular lymphocyte predominant. And this one actually have not too bad of a prognosis. So that's another beneficial thing. So this is the kind of concept that I want you guys to understand. The most common one is nodular sclerosis. The one with the poorest prognosis is lymphocyte depleted. The one with the best prognosis is lymphocyte rich. And all of these are under the umbrella of Reed-Sternberg cell positive cells, or CD1530 positive cells, or classical Hodgkin's lymphoma type. And the nodular lymphocyte predominant is on the umbrella of non-classical Hodgkin's lymphoma, CD2045 positive cells, or popcorn cells, which are Reed-Sternberg cell variants. Now, at this point, we have identified if it's Hodgkin's, if it's classical versus non-classical, and then what's the subtype, which can determine the degree of prognosis for the patient via histopathology. There's one more factor, and it's, did the actual Reed-Sternberg cells or the popcorn cells spread to other tissues? Did they have extranodal involvement? Are they really, really important? Are they involving multiple different lymph nodes? Are they spread everywhere around the body? How big are these lymph nodes? Because that's the next part. So then we go into staging. So when we talk about staging of Hodgkin's lymphoma, it's now going to the next part. So you see how we're kind of going in sequence of the lecture here? We started off with these Reed-Sternberg cells or popcorn cells accumulating in the lymph nodes. They cause lymphadenopathy, maybe some B symptoms, maybe some of the other like rare stuff like alcohol-induced pain, pruritus, Pell-Epstein fevers, and then maybe they have some splenomegaly. But the next part was they kind of spread. So if this leaches into the bloodstream, we said that they can spread. And they can go all over the dang place. They can go to the GIT, they can go to the liver, they can go to the spleen, they can go to the glands, they can go to the pleural and the pericardium, they can go to the brain, they can go to the bone marrow. These were all possibilities of where they can go. But we did say it's much less common in Hodgkin's as compared to non-Hodgkin's. Now, how would I be able to determine where these spread? Is there a way? Absolutely. There's two different tests that we can do. One is we can do what's called a PET-CT scan, and we can use something called a radioactive isotope called fluorodeoxyglucose. And what it does is cancer cells, especially these Hodgkin's lymphoma cells, they eat up this glucose. They consume it, and then it lights them up on the actual scan. And so it would light up which areas the actual neoplastic cells have invaded into. That's one test. An alternative could be a CT of the chest, abdomen, and pelvis with particularly IV contrast. This is another option because it'll basically kind of help to demarcate and highlight some of the masses in other particular tissues. So these are the alternative options for finding some of these masses that may have spread to other areas of the body, such as the actual GIT, the liver, the spleen, the glands, and the lungs and the heart. Now, if it's spread to the central nervous system, this is important. You want them to have neurosymptoms. If they have any neurosymptoms, or they're HIV positive, so if they have neurosymptoms, or they're HIV positive, you should probably test them to make sure it hasn't invaded the brain or the meninges. And so the ways that we can determine if it infiltrated the brain is we can get an MRI. The way that we can determine if it infiltrated into the meninges is testing the cerebrospinal fluid. So we can do a lumbar puncture. So those would be things that you want to consider, especially if the patient has any neurosymptoms or the HIV positive. You should definitely check an MRI to see if it invaded the brain, or do an LP to see if it invaded the meninges and subarachnoid space. And then lastly, what if it invaded the bone? If it invaded the bone, yeah, your CBC may have evidence of pancytopenia because it's invaded and kind of taken up a ton of space. But the other question is get a biopsy. If there's a lot of these actual Reed-Sternberg cells in the bone marrow, then we know it invaded there. So another option would be a bone marrow biopsy, and that'll confirm that you have Reed-Sternberg cells there. Okay? So these are the ways that you can go about doing that. And once you determine this, if any of these tests are positive, in other words, my bone marrow biopsy is positive for Reed-Sternberg cells, my MRI or my LP reveal, usually you have to do a combination of these because you need to confirm the actual tissue type. Maybe it'd be a biopsy or an LP. If any of these tests are positive, you have confirmed extranodal involvement. And that is really important because now that determines, okay, the Hodgkin's lymphoma is not just in the lymph nodes now, it's spread around the body. And that determines the degree of treatment and how aggressively we'll treat these patients. So that moves into the next part here, because we have to have an idea. In order for us to treat patients, we have to know how significant the tumor burden is in the body. And so what we do is we use a system called the Ann Arbor staging for Hodgkin's and non-Hodgkin's lymphoma. And what it does is we divide this into 4 stages. So we have stage 1, 2, 3, and you guessed it, stage 4. Now, for this Ann Arbor staging, what we do is we say, okay, if one of the lymph nodes, let's say it's just cervical, it could be cervical, supraclavicular, axillary. There's one lymph node that is involved here, all right? And we were able to determine that from a lymph node biopsy, that there was one lymph node involvement. That's stage 1. If there's two plus lymph nodes that were involved and they were on the same side of the diaphragm, so now I'm gonna put like this imaginary axis here. They're on the same side of the hemidiaphragm, that's stage 2. If I have 2 plus lymph node regions being involved, and they're on opposite sides, or both sides of the diaphragm, that's stage 3. And if I have any regions, I have 2 plus regions, whether it be, again, on opposite sides or on both sides, and I have a extranodal involvement. Let's just say, for example here, I pick the liver. I'm just picking the first one that I think of. There's extranodal involvement there. That is stage 4. So if you have 1 lymph node region, 2 lymph node regions, 2 plus lymph node regions on the same side of the diaphragm, 2 plus lymph node regions on both sides of the diaphragm, or extranodal involvement. So that's how we identify these. So one lymph node is positive, 2 lymph nodes positive on same side of diaphragm, 2 lymph node positive on both sides of the diaphragm. And then lastly, is there any extranodal positive involvement? Did it involve the pleura, the pericardium? Did it involve the brain? Did it involve the bone marrow? Did it involve what else? The liver? Did it involve the GIT, the glands? Technically, the spleen is what? In the most specific instances in the textbook, it's technically not an extranodal tissue. It's technically a nodal tissue. All right, so now all of this is technically a part of something called the Ann Arbor staging. So when you take into consideration all of this information, you get something called the Ann Arbor staging system. Now, you can actually add on to this Ann Arbor staging system with a sub-classification system. You're like, oh man, can't we just like make it simple? I promise it's not terrible. It consists of 3 sub-classifications. One is it's denoted by A, B, and X. Now, A is the absence of B symptoms. So this is based upon their clinical history. So is there negative B symptoms? If there's negative B symptoms, they get the actual subclassification A. If there is positive B symptoms, then they get the subclassification B. And if the lymph node tumor is approximately greater than or equal to 10 centimeters in diameter, then they get the subclassification system X. Now, with all of that being said, and this is also called bulky disease, is another way that you can describe the X. If you get a big old bulky lymph node mass, that's another indication. So with that being said, we start off with an example to kind of make sense of this. So here we have a patient, 2 lymph node regions are positive, or basically these big old swole lymph nodes, but they're on the right diaphragm side. So if we go back here, 1 lymph node, 2 lymph nodes on the same side of the diaphragm, that's stage 2. So far we can already write that down. But before we do that, let's make sure that there's no extranodal involvement. And B symptoms, with a lymph node diameter of approximately 15 centimeters, no mention of extranodal involvement. So because of that, we can already start this off with Ann Arbor stage 2. But what's the subclassification? Is there absence of B symptoms? No, there's presence of B symptoms. So that's B, so 2B, with a lymph node diameter of approximately 15 centimeters. That's greater than 10 centimeters. That's bulky disease. And so that is stage 2B, X. So that's how we would denote this Ann Arbor classification system of this patient's Hodgkin's lymphoma. At this point, we have covered how we've diagnosed Hodgkin's lymphoma. From here, we need to go into the next step, which is how do we treat Hodgkin's lymphoma? For stages 1 to 2, meaning one lymph node region, right, or two lymph node regions on the same side of the diaphragm, classical Hodgkin's lymphoma. So we know classical is Reed-Sternberg cell positive. If that's the case, we do something called ABVD therapy, and it goes adriamycin. Now, adriamycin is kind of like the brand name. It's technically called doxorubicin. This is what you actually should know it as, doxorubicin. The other one is called bleomycin. Now, the big thing here we'll talk about is a couple like pathophysiological, like downstream negativities from these drugs. We'll go over them in more detail when we do like chemotherapeutic agents like pharmacology, but we'll briefly talk about these in a second. The next one is vincristine. Vincristine, sometimes you see it written as Oncovin, so just be careful, you may see it written as Oncovin. And then the next one is called dacarbazine. So we have these particular agents that we would use in a patient who has Hodgkin's lymphoma, classical type, so this would be nodular sclerosis, lymphocyte predominant, or lymphocyte dominant, lymphocyte depleted, or some type of mixed cellularity, which was CD15-30 positive. Now, if they upgrade their stage to stages 3 to 4, so again, 2+ lymph node regions on both sides of the diaphragm, or extranodal tissue involvement, pleura, pericardium, brain, bone marrow, glands, salivary thyroid, or GIT, skin involvement, etc., they've upgraded to this stages. For this one, we can technically, if you really wanted to, you can do ABVD again times 6 cycles, or you could do Biacop. So this is bleomycin, this is etoposide, this is adriamycin, and again, adriamycin, just be aware that this is the brand name for the generic drug called doxorubicin. You could use cyclophosphamide, Oncovin, which is again the brand name for vincristine. The next one is called procarbazine, which is kind of like a form of dicarbazine. And then lastly is prednisone, but this is just to indicate that you could use any steroid. Prednisone, prednisolone, dexamethasone, it's just an indication that you want to use steroids. So at this point, if a patient comes in with Hodgkin's lymphoma, stage 1, 2, you do ABVD. Adriamycin, which is doxorubicin, bleomycin, or vincristine and dacarbazine. If they're stage 3 to 4, you can repeat the ABVD for 6 cycles or do the Biacop regimen. Bleomycin, etoposide, adriamycin, which is also known as doxorubicin, cyclophosphamide, then you have Oncovin, which is also known as vincristine, and then procarbazine, and lastly is any kind of steroid. We just put prednisone to make sense of the mnemonic. Now, big things to watch out for here that I want you guys to know is that bleomycin can cause a lot of pulmonary fibrosis. So when you're putting a patient on this drug, you need to make sure that you're monitoring their PFTs. Doxorubicin can cause a cardiomyopathy. So you want to make sure that you're monitoring these patients' echoes. So again, pulmonary function test for the pulmonary fibrosis from bleomycin. So again, you can see pulmonary fibrosis. So just make sure that you're monitoring those PFTs. And then doxorubicin can cause cardiomyopathy, like dilated cardiomyopathy. So make sure that you're monitoring the patient's echoes. All right, so for the non-classical Hodgkin's lymphoma, again, you have to remember that for these ones, this is that popcorn cell. So with the popcorn cell, you have to have those specific types of molecules on their surface. So they have to have, again, what were those molecules that we said? CD20 and 45. So CD20 and CD45 positive. If that's the case, and this really is these popcorn cells for the non-classical type, these will benefit more specifically from rituximab. And again, the most common subtype for this one was nodular lymphocyte predominant. Now the last one here is refractory CHL. So you've tried the ABVD, maybe you've done the Biacop regimen. After that, they're still not in remission. You can consider another drug, but they have to test positive. So this is for those Reed-Sternberg cells. So they have to be Reed-Sternberg cells. And on their surface, they have to have, so there was the CD15 and the CD30. you have to test positive for the CD30 molecule. If you test positive for the CD30 molecule, you can find potential benefit from a drug called brentuximab vedotin. And this is a type of monoclonal antibody that you can also utilize. So with that being said, in this video, we've talked about Hodgkin's lymphoma. We went over the pathophysiology. We went over all the different diagnostic tests. We went over how to treat these patients. I hope it makes sense. I hope that you guys enjoyed it. And as always, until next time.